Usually, when we think about building a defense system, there's this expectation of physical preparation, like a fortress.
Right. You build the walls before the attack.
Exactly. You see the enemy's battering ram coming, and you custom build a thicker door to block that specific weapon.
It's a very logical assumption. I mean, you expect biology to be reactionary, a direct physical response to a visible threat.
But immunology flips that entirely. It's wild. We're looking at a microscopic landscape where the body is prepping precise molecular defenses for pathogens it has literally never encountered before.
Never even seen.
Right. So today we are going to break down how the human immune system actually learns, how it remembers, how it regulates itself, and crucially, what happens when it fails.
Which is fascinating.
It really is. And for you listening, whether you are prepping for your medical boards, maybe working in a lab, or just an ambitious, self-taught learner, this deep dive is for you. We are drawing all our insights today from a really comprehensive medical text, the Illustrated Dictionary of Immunology, 2nd edition.
Yeah, by Dr. Julius M. Cruse and Dr. Robert E. Lewis. It's a text that synthesizes decades of complex molecular biology, pathology, and clinical diagnostics.
It's dense.
Well, very dense. But our mission today is to translate those dense immunological mechanisms into clear, practical knowledge that you can actually apply in the clinic or the lab.
Perfect. So before we can even begin to understand how to manipulate the immune system in a clinical setting, we have to understand how the body actually produces its specific defenses. Because historically, science had this completely backwards.
They really did. For a long time, the leading idea was the instructive theory, or some people called it the template theory.
Okay, let's unpack this. What was the template theory?
Well, the concept was that when a foreign antigen, so like a virus or a bacteria, entered the body, the immune system's uncommitted proteins would physically encounter the invader and fold themselves around it.
Like clay molding to a statue.
Exactly like that.
Which goes right back to my fortress analogy. The idea that the body sees the shape of the battering ram and custom forges a door on the spot.
Right.
And it makes intuitive sense, but it falls apart the second you think about immunological memory. I mean, if the antigen is the physical template, what happens when the antigen is destroyed and cleared from the body?
That is the glaring flaw.
Yeah. How does the body remember how to make that specific antibody years later when the mold is literally gone?
It can't. It just can't. Furthermore, as biochemistry advanced, we learned that antibodies are synthesized based on highly specific genetic amino acid sequences.
They aren't just squishy blobs of protein.
Right. They can't be remolded at will. The sequence dictates the shape. End of story. So the instructive theory had to be completely abandoned in favor of selective theories.
Which brings us to Paul Ehrlich.
Yes. Ehrlich had his side chain theory back in 1900, but the modern accepted framework we use today is the clonal selection theory.
Proposed by Burnett and Talmage in the 50s, right?
Exactly. This theory postulates that lymphocytes, your B and T cells, are genetically programmed to react to a specific antigen prior to ever being exposed to it.
So to visualize this for the listener, think of a locksmith. The old template theory thought the body looked at an invading lock and forged a brand new key on the spot in real time.
Right, but the clonal selection theory says the body actually has a giant wall of like a billion randomly shaped, already made keys.
It just tries them all until one fits the invader, and then it mass produces that specific key.
Yes, exactly. Though biologically, those keys are antigen receptors sitting on the surface of patrolling lymphocytes. And what's fascinating here is we actually prove that this vast array of pre-made keys exists through a mathematical breakthrough.
Oh, the Wukobat plot.
Yes, detailed right there in the text.
I want to dig into that because how do you mathematically prove a biological structure before we had modern supercomputers mapping genomes?
It was incredibly clever. Researchers took the amino acid sequences of many different antibodies and just aligned them. They calculated an index of variability, basically a mathematical score, for every single amino acid position along the antibody chain.
Okay, so tracking how much they change.
Right. If the template theory were true, and proteins just sort of folded around antigens, you'd expect the variability in the amino acids to be spread out.
Just somewhat evenly across the whole molecule.
Exactly, because the whole molecule is just shifting around to fit whatever mold it encounters.
But that's not what they found.
Not at all. The Wukobat plot graphically proved that the extreme variability, the unique, custom-fit parts of the antibody, are tightly clustered.
In the hypervariable regions.
Yes, 3 very specific locations, The rest of the antibody is highly conserved and stable.
Oh, wow.
So this proved definitively that the genetic sequence is dictating a highly specific, predetermined binding site. The wall of keys is real, and it is genetically encoded before the lock ever shows up.
So the body encounters an antigen, tests its patrolling lymphocytes, and finally finds the one single cell with the exact right hypervariable receptor. But, I mean, one cell can't fight off a systemic infection.
No, it would be overwhelmed instantly. So
Wow.
once that specific lymphocyte is selected by the antigen binding to it, it undergoes clonal expansion.
It builds the army.
Exactly. It rapidly proliferates, creating thousands of identical clones of itself, all bearing that exact same specific receptor. These clones then differentiate into effector cells.
Like plasma cells pumping out antibodies.
Right, to clear the pathogen.
And this leads us to the anamnestic response, which is just the clinical term for immunological memory.
Yes.
So the first time your body sees a pathogen, it mounts a primary response. This takes time, right? Like days or weeks, because you have to find that one specific lymphocyte and multiply it.
And this primary response is predominantly driven by immunoglobulin M, or IgM antibodies.
But once the pathogen is cleared, a portion of those clone cells remain in the system as memory cells.
Right, they stick around. So
Yeah.
if that exact same antigen ever returns, you don't have to start the search from scratch. The secondary response, the anamnestic response, is incredibly fast.
And of a massively greater magnitude.
Massively.
Yeah.
Because those memory cells are already primed.
Ah.
And the secondary response is primarily driven by IgG antibodies.
Here's where it gets really interesting, though, because as a learner, you're taught that this memory mechanism is universally a good thing. I mean, it's the whole basis of vaccination.
Usually, yes.
But the text brings up a phenomenon called original antigenic sin, where immunological memory actually sabotages the patient. Why does the immune system get stuck in the past?
Original antigenic sin is a critical concept, especially when you are dealing with rapidly mutating pathogens like the influenza virus.
Okay, walk us through it.
Imagine you get infected with a specific strain of the flu. Your body mounts a primary response and creates memory cells against the specific surface markers, the epitopes of that virus.
And then you recover.
Right, you recover. But then, say 5 years later, you are infected with a new strain of the flu. It's similar, but it has mutated and presents new epitopes.
Logically, my immune system should recognize the new epitopes, activate naive, uncommitted lymphocytes, and make new, highly specific antibodies.
But it doesn't.
Wait, really?
It doesn't. Instead, your immune system stubbornly mounts a massive secondary response using the old memory cells, targeting only the original epitopes it remembers.
While completely ignoring the new, highly immunogenic epitopes on the current virus.
Exactly. It's fighting the last war.
But why would biology allow this massive blind spot? If memory is a survival advantage, why ignore the new weapons?
It comes down to kinetics and resource competition. Memory NE and T cells have receptors with a much higher affinity for the antigen, and they exist in vastly higher numbers than any single naive cell.
So they just outcompete them.
Exactly. When the mutated virus enters, those memory cells just swarm it. They bind to whatever original epitopes are left, sucking up all the antigen and all the chemical survival signals.
Before the naive cells can even get activated to learn the new epitopes.
Right. It is an evolutionary gamble. Most of the time, this rapid suppression of a returning pathogen saves your life. But with a virus that uses antigenic drift to constantly change its code, that resource monopoly totally backfires.
That is wild. Okay, so we have an immune system that can mass produce armies of clones. It has a hair trigger memory, and it monopolizes resources to brutally attack anything it recognizes.
It's formidable.
So how does this heavily armed system avoid destroying the host's own tissue?
Through immunologic tolerance. And this isn't just a passive ignoring of self,
Yeah.
It is an active, carefully regulated process. And the kinetics are fascinating.
Yeah, the mechanisms for silencing T cells are vastly different from silencing B cells, according to the text.
They're very different.
It notes that T cell tolerance is incredibly fast to induce. It takes just 1 day, and it lasts up to 150 days. But B cell tolerance takes about 10 days to induce. It requires 100 times more of the antigen inducing the tolerance, and it only lasts 50 to 60 days.
Right.
Why the massive discrepancy?
Think about the hierarchy of the immune response. T cells, specifically helper T cells, act as the generals of the immune system. They secrete the cytokines that coordinate the rest of the attack.
Yep.
If you tolerize the generals quickly, the foot soldiers, the B cells, cannot mount a coordinated, sustained attack anyway. Biology designed the system to shut down the command structure first.
Oh, that makes perfect sense. And B cells are the weapons factories pumping out antibodies. Why make them harder to tolerize?
Because you want those factories to be robust. During a massive systemic infection, you are flooded with foreign antigen. If B cells were easily tolerized by high doses of antigen, your immune system would shut down right when the infection is at its absolute worst.
Ah, so B cells require a much higher threshold of proof.
Yes, 100 times more tolerant before they stand down.
So how do they actually stand down?
Well, in immature B cells, encountering massive self-antigen, they undergo what's called clonal abortion. They are just deleted before they can fully develop.
And in mature B cells?
We see cloning exhaustion.
Becker
If
error.
there's a massive overwhelming presence of an antigen, it forces the B cell to continuously proliferate and secrete antibodies until it literally runs out of intracellular machinery and resources.
Oh, wow, it burns itself out and dies.
Precisely.
So tolerance handles the cells that might target our own tissue. But what about when fighting a real invader? If you have this fork in the road where the body can either launch a humoral response making antibodies or a cellular response sending in cytotoxic T cells, how does it choose?
Right, without causing a chaotic traffic jam of conflicting signals.
Exactly.
It uses immune deviation, which is driven by macrophages at the site of infection. Depending on the nature of the pathogen, say an intracellular virus versus an extracellular bacteria, the macrophage will secrete specific cytokines to push the immune system down one path.
The text highlights interleukin 12 and interleukin 4 here.
Right. If the macrophage secretes IL-12, this triggers the development of Th1 cells, which drive the cellular immune response.
And if the environment favors IL-4.
Then this triggers Th2 cells, which drive antibody synthesis. And the critical mechanism here is cross-inhibition.
Meaning they block each other.
Yes, the cytokines produced by Th1 cells actively suppress the Th2 pathway and vice versa. It forces the immune system to commit to one coordinated strategy.
It prevents the body from throwing every weapon at the wall simultaneously, which would lead to catastrophic systemic inflammation.
Exactly.
But once the pathogen is destroyed, how does the system finally turn off? The text introduces Niels Journey's network theory, which is notoriously difficult to conceptualize.
It is a profound concept. Jern proposed that the immune system regulates itself by recognizing its own antibodies as foreign.
Okay, let me try to break this down step by step with an analogy. Imagine the immune system is a hall of mirrors.
I like this. Go on.
You create a highly specific antibody to attack a virus. Let's call it antibody 1.
Yeah.
But antibody 1 is so custom fit, its own binding site, its idiotype, has a unique physical shape that has never existed in the body before.
Correct. So to the rest of the immune system, the binding site of antibody 1 looks like a new foreign antigen.
So the body reacts by creating antibody 2 to attack the binding site of antibody 1. But if antibody 2 physically fits perfectly into the lock of antibody 1, then the tip of antibody 2 must physically resemble the shape of the original virus.
Yes. By creating this anti-idiotype antibody, antibody 2, the immune system has generated an internal structural reflection of the original invader. This is the internal image.
So what does that do?
As antibody 1 clears the actual virus, antibody 2 begins to bind to antibody 1. This saturates the receptors on the B cells. The B cells receive a signal that all their receptors are bound up, which tells them the war is over.
It's time to downregulate the response.
Exactly. Without this internal image network channeling the expansion, the primary response would be a perpetual runaway train.
That is incredible. Okay, so what does this all mean? Let's transition from molecular theory to clinical practice. We have to apply this to the medical laboratory and pathology.
Absolutely.
How do we exploit these mechanisms for diagnostics, and what does it look like when they fail in a patient?
Let's look at diagnostics first, specifically the Coombs test, which is a total staple in hematology. We use it to detect autoantibodies on a patient's red blood cells.
The problem is that even if a patient has IgG autoantibodies attacking their red blood cells, you can't just look at the blood under a microscope and see it.
No, you can't, because red blood cells have a negative surface charge, creating what's called the zeta potential.
Zeta potential.
Right. This charge makes the cells repel each other, keeping them suspended in the plasma. Now, IgG antibodies are relatively small. Even if they are coating the red blood cells, they aren't physically long enough to bridge the gap between two repelling red blood cells and bind them together.
So the cells don't clump.
Exactly, no clumping.
So how do we force a visible diagnosis in the lab?
We use the immune system's own binding logic. We take rabbit serum containing antibodies specifically designed to bind to the constant region of human IgG.
So an anti-antibody.
Yes. We add this rabbit anti-antibody to the patient's blood. The rabbit antibodies are large enough to act as bridges. grabbing an IgG on one red blood cell and an IgG on another.
Oh, I see.
This overcomes the zeta potential, causing the red blood cells to physically clump together, which is agglutination, which we can easily see in the lab.
And we use similar homing logic in immunocytochemistry, too. Pathologists take a specific antibody, tag it with a fluorescent dye called a fluorochrome, and wash it over a tissue biopsy.
Right.
If the target molecule, say a specific cancer protein, is present, the antibody binds, and that specific cellular structure literally glows under a fluorescence microscope.
It's turning molecular specificity into a visual map. It's brilliant. But we also have to look at what happens when these specific systems turn against the patient.
Yeah, pathology.
Consider idiopathic thrombocytopenic purpura, or ITP.
This is an autoimmune failure targeting platelets, the cells responsible for blood clotting.
In ITP, the patient's body produces IgG autoantibodies that mistakenly coat their own platelets. But the antibodies themselves don't actually destroy the platelets.
Oh, they don't.
No. They act as molecular tags. They basically signal for mononuclear phagocytes, specifically macrophages living in the spleen, to hunt them down.
Right, to consume them. Connecting back to what we discussed earlier, these are the same family of cells that dictate immune deviation, but here they are carrying out a destructive order.
Exactly. The spleen destroys these coated platelets vastly faster than the bone marrow can replace them. The result is severe thrombocytopenia, leading to purple bruising beneath the skin and dangerous, uncontrolled bleeding.
Man, there is another pathology detailed in the text that bridges everything we've talked about, hyper-IgE syndrome, or HIE.
Yes, HIE.
The clinical presentation involves severe eczema and recurrent cold abscesses, typically caused by Staphylococcus aureus.
And the defining laboratory finding in HIE is a massive pathological elevation in immunoglobulin E, or IgE. We're talking levels greater than 5,000 international units per milliliter.
For anyone studying this, understanding why an abscess is cold in HIE isn't just trivia to memorize. It directly correlates to the molecular structure. Normally, a staph abscess is hot, red, and swollen. Why is it cold here?
Because the massive immune response is dominated by the wrong class of antibody. Structurally, IgE cannot fix complement.
The complement system being the protein cascade that punctures bacterial walls and calls in neutrophils, causing massive inflammation, heat, and swelling.
Exactly. Without complement activation, that fiery inflammatory cascade never triggers. The staph infection just stews in the tissue without generating heat, hence a cold abscess.
Wow.
But the pathology of HIE cascades far beyond localized skin infections.
Yeah, the text details a mechanism where this antibody defect actually leads to bone loss, which sounds like an enormous leap.
It's a perfect example of how interconnected the network is. The body recognizes this massive excess of IgE and tries to regulate it by producing IgG antibodies against the IgE.
Just like Jern's network theory, it's trying to bind up the excess.
But it backfires. These IgG immune complexes circulate and bind to those mononuclear phagocytes we just talked about.
The macrophages.
Right. This inappropriate binding triggers the macrophages to release specific chemokines signaling proteins. These chemokines travel through the blood and act on osteoclasts, the cells that break down bone, inducing severe calcium resorption.
So an isolated molecular defect, the overproduction of IgE cascades into immune complexes, which trigger macrophages, which dump cytokines, which literally instruct the body to eat its own bones.
Exactly.
That is why understanding the underlying mechanism is so much more critical than just memorizing a list of symptoms.
It all comes back to causality.
All right, to wrap up our mission today, we're going to reinforce what you've learned with a rapid-fire clinical vignette for you, the listener. Ready?
Let's do it.
If your patient is infected with a new mutated strain of a virus, but their immune system stubbornly monopolizes resources to produce massive amounts of antibodies tailored only to the specific strain they caught 5 years ago, what immunological concept is sabotaging their recovery? I'll give you a second.
Think about the flu.
If you said original antigenic sin, you nailed it.
And as we close, I want to point you toward a frontier mentioned in the text that completely redefines how we interact with this system: genetic immunization. We spent today talking about how the immune system reacts to threats, and historically we use dead or weakened pathogens to train it.
But genetic immunization skips the pathogen entirely.
Completely. Instead of injecting a patient with a piece of a virus, researchers can inject naked plasmid DNA straight into the muscle tissue. That DNA encodes the blueprint for the viral protein.
Okay.
Our own muscle cells take up that DNA and start manufacturing the pathogen's protein themselves.
We turn the human body into its own vaccine factory.
The immune system then spots these foreign proteins being produced locally and mounts a full adaptive response, generating memory cells without ever seeing the actual bug.
That is incredible.
It bypasses all the old methods of cultivation and opens up an entirely new frontier for how we might treat infectious diseases, cancers, and even autoimmune disorders. Something definitely worth exploring on your own.
So we started today thinking you had to look at the battering ram to build the door. But as we've seen, the immune system already has a billion doors waiting. And with genetic immunization, we are learning how to hand the body the blueprints so it can build the training dummies itself.
It's not a reactionary fortress at all.
No, it's a predictive, highly regulated survival engine. Keep studying the mechanisms, keep asking why, and we'll catch you on the next deep dive.